Hello students, welcome to EPG Pathshala. Myself Dr. Sheetal Shirodkar. I am working as an assistant professor in Amity Institute of Biotechnology, Amity University, Noida. Today we will be presenting a lecture on the topic Prokaryotic Translation from the paper Molecular Biology, Genetic Engineering and Biotechnology. The objectives of our study will include understanding the concept of genetic code, understanding the concept of Wobble Hypothesis, explain the process of prokaryotic translation, explain about different types of post-translational modifications in prokaryotes. The prokaryotic translation process can be divided into 5 main stages: activation of amino acids, initiation, elongation, termination and post-translational modifications. Translation or protein synthesis is a central process of central dogma of molecular biology. It deals with production of proteins or chains of amino acids by making use of messenger RNA molecule as a template, ribosomes as protein synthesizing machinery and transfer RNA molecules as carriers of amino acid during the translation process. Living cells devote about 90% of their chemical energy to the synthesis of proteins and only about 10% to the other biosynthetic processes. More than 35% of the dry weight of the cell consists of ribosomes, proteins involved in translation and transfer RNA molecules. This suggests that protein synthesis is an important process for the survival of microorganisms. Protein synthesis process in E. coli occurs at a very high polymerization rate. About 100 amino acids are polymerized in 5 seconds. It is also a highly regulated process so that enough proteins are made to meet up the cellular requirements. Along with synthesis, targeting and protein degradation process ensures optimal cellular concentration of the proteins. The prokaryotic translation process can be divided into 5 main stages, which includes activation of amino acids, initiation, elongation, termination and post-translational modification processes. Coming to the schematic representation of prokaryotic translation, the process starts with the initiation complex. The 70S initiation complex of prokaryotes contains 3 binding sites for transfer RNA molecules: the P site for the peptidal transfer RNA binding, the A site for the amino acyl transfer RNA binding and the E site or the exit site from where the uncharged transfer RNA leaves. Apart from that, the 70S ribosome binds the messenger RNA molecule. The messenger RNA molecule during the initiation binds to the precise location so that the AUG codon is present at the P site of the ribosomes. The formilated methionine transfer RNA is the first transfer RNA that binds to the AUG codon or the ribosomal P site. Once the initiating complex is formed, the ribosomes then enter into the elongation stage. The subsequent amino acyl transfer RNAs bind to the A site of the 70S ribosome. The 70S initiating complex then enters into the elongation stage of translation. The amino acyl transfer RNA EFTU GTP complex enters and binds to the ribosomal A site. Simultaneously, GTP is hydrolyzed and EFTU GDP complex is released. The next step is peptidal transferase reaction, during which peptide bond formation takes place between the amino acids linked to their respective transfer RNA molecules. During peptidal transferase reaction, peptide bond formation takes place between the amino acids linked by their respective transfer RNAs to the ribosomal P and the A site. Following peptide bond formation, we now have a diacyl transfer RNA present at the ribosomal A site and a deacylated transfer RNA present at the ribosomal P site. Next, in presence of elongation factor G GTP complex, ribosome moves by a distance of 1 codon towards the 3' end of the messenger RNA molecule. This results in formation of a diacylated transfer RNA at the ribosomal P site and a deacylated transfer RNA at the ribosomal E site. The deacylated transfer RNA now leaves the complex and is recycled. The process continues till a termination codon is present at the ribosomal A site, following which the release factors bind to the ribosomal A site and the process is terminated. Next we come to the components of translation. The different components of translation process includes messenger RNA, which acts as a template for the protein synthesis process and is read in 5' to 3' direction. Transfer RNA molecule, which acts as an adapter or the carrier of amino acids. Ribosomes, which are the protein synthesizing machinery. Enzymes, which are required for attachment of amino acids to the correct transfer RNA molecule and for peptide bond formation between amino acids. Various soluble proteins are also required for proper initiation, elongation and termination of translation. Structure of transfer RNA molecules. Transfer RNA molecules, they act as adapters or carriers of amino acids. It has a clover leaf-like structure. The amino acids are attached to its 3' hydroxyl end. It also contains an anticodon arm that carries anticodons. These are important to bind to the specific codons on the messenger RNA molecule during the translation process. The anticodon sequence is complementary to the codon for the amino acid. For example, the codon for alanine is GCA, read in 5' to 3' direction, and the anticodon for it is CGU, read in 3' to 5' direction. Next we will discuss about the components of the prokaryotic ribosomes. Ribosomes are the protein synthesizing machinery. It recognizes the signal sequences that allows initiation of the protein synthesis process. It also ensures accuracy of the protein synthesis process by stabilizing the transfer RNA messenger RNA interaction. It is involved in enzymatically linking the amino acids and formation of the polypeptide chain. They move along the messenger RNA molecules as they read through the messenger RNA in 5' to 3' direction. The 70S prokaryotic ribosome is made up of the smaller 30S subunit and the larger 50S subunit. The smaller 30S subunit is made up of 16S rRNA molecule and 21 different proteins, while the larger 50S subunit is made up of 23S rRNA, 5S rRNA and about 34 different proteins. Enzymes involved in protein synthesis process. There are two major enzymes that are involved in the protein synthesis process. First is the amino acyl transfer RNA synthetase. This enzyme catalyzes attachment of the transfer RNA molecule to its corresponding amino acid. There are 20 different amino acyl transfer RNA synthetases, one for each amino acid. The attachment of the amino acid charges the transfer RNA molecule. Peptidyl transferase. Peptidyl transferase enzyme catalyzes peptide bond formation between the amino acids linked by their transfer RNA to the ribosomal P site and A site. They allow sequential transfer of amino acids to the growing chain. Figure number 4. Now we will discuss about genetic code and its properties. A set of 3 bases forms one codon or a genetic code. A total number of 64 codons, 61 sense codons and 3 termination codons are present. Genetic code is a triplet. Genetic code is degenerate, meaning that a particular amino acid can be coded by more than one triplet codons. For example, lysine amino acid has two-fold degeneracy, meaning that it is encoded by codons AAA and AAG. Genetic code is non-overlapping in nature. As you can see, the first codon is CAT while the second codon is TGA. Thus, the base T is shared between the two codons. This is an example of an overlapping codon. However, this is not the case with genetic code, and the codons are actually non-overlapping in nature. Genetic code is commaless, which means that no comma or no punctuation is required to indicate the end of one codon and the beginning of the next codon. Genetic code is non-ambiguous. This means that the same codon does not codes for more than one amino acid. Genetic code is universal, which means the same code AUG, if it codes for amino acid methionine in microbes, then it will also code for methionine in other living systems including plants and animals. Genetic code has polarity. It means that it is always read in 5' to 3' direction. Initiation codon, the triplet codon AUG codes for the initiation codon. The initiating AUG codon codes for formilated methionine residues, while all the other internal AUG codons code for unformilated methionine residues. Chain termination codons, there are in all 3 termination codons UAA, UAG and UGA. Next we will discuss about the concept of Wobble hypothesis. Wobble hypothesis explains about the multiple recognition of codons by a transfer RNA molecules. According to Wobble hypothesis, the first 2 bases of the messenger RNA codon pairs with the corresponding base of the anticodon very specifically. The first base of the anticodon is read in 5' to 3' direction and determines the number of codons recognized by the transfer RNA. When an amino acid is specified by several different codons, the codons that differ in either of the first 2 bases require different transfer RNAs. A minimum of 32 transfer RNAs are required to translate all 61 codons, 31 to encode the amino acids and 1 for the initiation. Now we will discuss in more details about the different steps of prokaryotic translation process. As already explained, the prokaryotic translation can be divided into 5 main stages: activation of amino acids, initiation, elongation, termination and release, and post-translational modifications. The first stage is activation of amino acids. During activation of amino acids, 20 different amino acids, 20 different amino acid transfer RNA synthetases, 32 or more transfer RNA molecules, ATP and Mg2+ ions are required. Amino acid transfer RNA synthetases catalyzes attachment of an amino acid to the corresponding transfer RNA molecule. This reaction takes place in the cytosol and requires Mg2+ ions as cofactors and energy from ATP hydrolysis. Now we will discuss about the different steps of initiation process. The components required for prokaryotic initiation process includes 30S ribosomal subunit, the coding messenger RNA sequence, the initiating formilated methionine transfer RNA FMET, 3 initiating factors IF1, 2 and 3, GTP as energy source, the 50S ribosomal subunit and Mg2+ ions as cofactors. The different steps of initiation are as represented in the figure 8. During the first step of initiation process, the 30S ribosomal subunit binds to the 2 initiation factors, initiation factor 1 and 3. Initiation factor 3 prevents premature association of the 30S and the 50S ribosomal subunits. The messenger RNA then binds precisely to the 30S ribosomal subunit. such that the 5' AUG codon is placed at the ribosomal P site. The initiating AUG is directed to the appropriate location because of its close proximity to the Shine-Dalgarno sequence. This consensus sequence consists of 4 to 9 purine bases placed approximately 8 to 13 base pairs before the 5' end of the initiation codon. The 16S rRNA of the 30S ribosomal subunit contains a pyrimidine-rich series of bases near its 3' end, which is complementary and binds specifically to the Shine-Dalgarno sequence, allowing correct positioning of the 5' AUG codon. The Shine-Dalgarno sequence forms complementary base pairing with the pyrimidine-rich series of bases towards the 3' end of the 16S ribosomal RNA of the 30S ribosomal subunit. The bacterial ribosomes contain 3 binding sites for amino acyl transfer RNA, the A site, the P site and the E site that binds newly incoming amino acyl transfer RNA, peptidal transfer RNA and deacylated transfer RNA respectively. The initiating 5' AUG positions itself correctly to the ribosomal P site, which binds with the formilated methionine transfer RNA F-met. The subsequent incoming amino acyl transfer RNA of the translation process binds first to the A site. The E site or the exit site is the location from where the uncharged transfer RNA leaves following peptide bond formation during elongation. In the next step of initiation, a complex consisting of GTP bound initiation factor 2 and formilated methionine transfer RNA F-met associates with the complex of ribosomal 30S subunit, initiation factor 1, initiation factor 3 and messenger RNA. The anticodon of the transfer RNA pairs correctly with the initiation AUG positioned at the ribosomal P site. In the third step, the ribosomal 50S subunit now associates with the complex, which is accomplished with GTP hydrolysis into GDP and pyrophosphate along with release of 3 initiating factors. This results in functional 70S initiation complex associated with the messenger RNA and the initiating formilated methionine transfer RNA F-met. This is now ready to continue with the elongation steps. 3 different interactions ensure proper binding of the initiating formilated methionine transfer RNA F-met to the ribosomal P sites. The codon-anticodon interaction between the formilated methionine transfer RNA F-met and the initiating AUG codon, the interaction between the Shine-Dalgarno sequence and the 16S rRNA ribosomal sequence, and the binding interactions between the formilated methionine transfer RNA F-met and the ribosomal P site. The initiation stage. The initiation codon is AUG. It is towards the 5' end of the messenger RNA molecule that is being translated. However, it is not the first 3 nucleotides of the messenger RNA molecule. It also determines the reading frame. In prokaryotes, there is a conserved region about 7 nucleotides which is upstream of the initiating AUG codon. This region contains 6 nucleotide sequence which is known as the Shine-Dalgarno sequence. The Shine-Dalgarno sequence reads AGG, AGG in 5' to 3' direction. This sequence is complementary to the region at the 3' end of the 16S rRNA of the small subunit. Base pairing between the complementary sequences stabilizes the binding of the small ribosomal subunit to the messenger RNA for proper assembly. In prokaryotes, the first AUG is recognized by a special transfer RNA known as transfer RNA F-met carrying a modified methionine residue. It is also known as the formilated methionine transfer RNA. Three initiation factors, initiation factor 1, 2 and 3 are required for the process. The 30S ribosomal subunit is first bound by initiation factor 1 and 3. Following binding of the two initiation factors, the 30S ribosomal subunit undergoes conformational change and then interacts with the messenger RNA molecule. The messenger RNA molecule starts scanning the ribosomes until there is precise pairing between the. We will now discuss figure 8. We will now discuss in more details about the different steps of prokaryotic initiation process. Three initiating factors, initiation factor 1, 2 and 3 are involved in translation initiation in prokaryotes. The 30S ribosomal subunit is first bound by initiation factor 3 and initiation factor 1. This brings about conformational change in the ribosomal 30S subunit and is now ready to bind with the messenger RNA molecule. The messenger RNA molecule begins to scan the 30S ribosome and binds precisely so that the AUG codon is now at the ribosomal P site. The initiating transfer RNA methionine bound to initiation factor 2 and GTP now enters and binds to the ribosomal P site. The ribosomal 50S subunit now enters. Simultaneously with it, GTP hydrolysis occurs and the three initiating factors are released. This results in formation of a functional initiation complex, which is the 70S initiation complex that is bound with the initiating methionine transfer RNA to the ribosomal P site. The initiation complex is now ready for the elongation cycle. The requirements for the prokaryotic translation elongation includes the initiation complex, amino acid transfer RNAs, a set of three soluble cytosolic elongation factors EFTU, EFTS and EFG and GTP. During the first step of elongation, the next amino acid transfer RNA binds to the ribosomal A site. The amino acid transfer RNA enters in the form of a complex with elongation factor TU and GTP. Thus, a complex of amino acid transfer RNA EFTU GTP binds precisely to the ribosomal A site. Hydrolysis of GTP takes place and elongation factor TU GDP complex is released from the 70S ribosomes. The elongation factor TU GTP complex is regenerated in a process requiring elongation factor TS and GTP. In the second step of elongation cycle, peptide bond formation takes place amongst the amino acids attached to the ribosomal P and A site by their respective transfer RNA molecules. During this step, the amino group of the amino acid at ribosomal A site makes a nucleophilic attack and displaces the transfer RNA in the P site, resulting in peptide bond formation between the two amino acids. This results in formation of a dipeptidyl transfer RNA at the A site of the ribosomes and a deacylated transfer RNA at the P site of the ribosomes. The reaction is catalyzed by enzyme peptidyl transferase, which is now known to be catalyzed by the ribosomal 23S rRNA subunit of the ribosomes. Translocation is the third step of elongation, during which the ribosome moves by a distance of 1 codon towards the 3' direction of the messenger RNA. This results in repositioning of the dipeptidyl transfer RNA to the P site of ribosomes and the deacylated transfer RNA to the E site of ribosomes, from where the transfer RNA is released into the cytosol. The reaction is catalyzed by enzyme translocase or elongation factor G in a reaction requiring energy from GTP hydrolysis. The third codon is now at the ribosomal A site and the ribosomes are ready for the next elongation cycle. For each amino acid molecule added to the growing polypeptide chain, 2 GTP molecules are hydrolyzed into GDP and pyrophosphate. Prokaryotic translation termination. The elongation process continues until the ribosome encounters a termination codon on the messenger RNA molecule. There are 3 codons that signal translation termination. These include UAA, UAG and UGA. Presence of one of these codons after the final coded amino acid acts as a translation termination signal. In bacteria, when the termination codons are positioned at the A site of the ribosomes, the termination factors or the release factors RF1, RF2 and RF3 carry out breakdown of the bond between the polypeptide and the transfer RNA molecule. This results in release of free polypeptide and the transfer RNA molecule. The 70S ribosomal subunit then dissociates into 30S and 50S subunits to begin with a new protein synthesis cycle. Post-translational modifications. Folding and processing of the newly synthesized polypeptide chain takes place during the post-translational modifications. After synthesis, the newly synthesized polypeptide chain assumes its native conformation that is guided by a number of interactions like hydrogen bonds, van der Waals interactions, ionic bonds and hydrophobic interactions. Some of the polypeptides, however, undergo additional processing or post-translational changes. Some of these post-translational modifications are discussed here. The first modification includes modification of amino terminal and carboxy terminal amino acids. In bacteria, the first amino acid in all polypeptide chains is an N-formyl methionine residue. The formyl group and the initiating methionine and sometimes further amino terminal and carboxy terminal amino acids are enzymatically removed from the final protein structure. The second kind of modification is loss of signal sequences. Signal sequences range from 15 to 30 residues in length and they play an important role in targeting the proteins to their final destination. Once the protein has reached its final location, the signal sequences are enzymatically cleaved and they are not part of the final functional protein. Modification of individual amino acids. Bacteria modify several amino acid side chains of the proteins by addition and removal of phosphate groups by enzymes named kinases and phosphatases respectively. In bacteria, kinases often phosphorylate histidine and aspartate residues, which are important modifications in bacterial two-component regulatory system. Example as shown in the figure consists of a two-component system made up of a sensor protein which contains a histidine kinase domain that autophosphorylates the histidine amino acid in response to a signal. The kinase then transfers the acquired phosphate residue to an aspartate residue of the second component, which is known as the response regulator. The phosphorylated regulator then controls transcription of several downstream genes, thus operating a regulatory cascade. Protein glycosylation in bacteria is another kind of post-translational modification. Many surface appendages proteins like pillin of pili and flagella of flagellin contain glycosylated serine and threonine residues. The process has been described in both gram-positive and gram-negative bacteria and may play an important role in adhesion, stabilization of the proteins against proteolysis, as well as evasion of host immune system. The commonly glycosylated amino acid residues include serine and threonine residues which are O-glycosylated and asparagine residues which are N-glycosylated. Next modification includes addition of prosthetic groups. Prosthetic groups are non-protein parts of some proteins that are required for their activity. Prosthetic group may be organic or inorganic in nature but is never made up of amino acids. They are tightly bound to the protein component through covalent bonds. For example, ferredoxins are a family of bacterial proteins containing 2, 4 or 8 atoms of iron and additional inorganic sulfate. Next type of post-translational modification includes proteolytic processing. Many proteins are produced as large inactive form of proteins which are known as precursors. Later during the post-translational modifications, they are proteolytically cleaved into smaller active forms of the proteins. Many bacterial toxins achieve their high potency by delivery of catalytically active polypeptide fragment of the toxin to the eukaryotic cell cytosol. Activation occurs by proteolytic cleavage of the polypeptide at the defined sites. Examples include toxins like diphtheria toxin, anthrax toxin, etc. The last kind of post-translational modification includes formation of disulfide cross-links. Post-translationally, many proteins fold into its native conformation by formation of intrachain or interchain disulfide bonds between their cysteine residues. These bonds are sometimes important in formation of the functional active protein molecule. For example, enzyme alkaline phosphatase from E. coli is a homodimer wherein 2 intramolecular disulfide bonds are involved in formation of the dimeric protein with full enzymatic activity. Thus, in summary, in this lecture we have learnt about genetic code and its properties, the concept of Wobble hypothesis, steps in prokaryotic translation process, and different types of prokaryotic post-translational modifications. Thank you. You can also find this lecture on the UGC ePathshala website.